This is the lesson for transcription. Transcription is the process of making an RNA molecule from a DNA molecule. So what is the overview of transcription? Obviously, you are making a copy of the DNA instructions into RNA. Both are nucleotides, so it's a very straightforward transfer of information from DNA to RNA. Now, because it's straightforward, some may ask, well, why even bother doing it, which we'll address in just a moment. But the purpose then of the RNA molecule that you're then going to make is the RNA is then going to be used to make proteins. Now, not all of the DNA is going to be used to carry out transcription at a time. This is one of the important differences between transcription and replication. Replication for DNA involves all the DNA. Transcription only involves a small portion of the DNA which you make a copy of into an RNA form. That part of the DNA that's used to make RNA is called a transcription unit. Now back to the question, why? If you already have instructions in the form of DNA, and RNA is a very similar molecule, both made out of nucleotides, some slight differences obviously, your cell in RNA, thymine in DNA, but otherwise they're very similar, so why not just use the instructions in DNA? Well, one, the DNA is in the nucleus and is protected in the nucleus. Protein synthesis, which is the purpose of making an RNA molecule, is so that eventually you can use it to make protein. That process, protein synthesis or translation, occurs in the cytoplasm. If you were to use DNA, you would then suddenly have to have the DNA leave the nucleus and enter the cytoplasm where translation can happen. One, this leaves the DNA open for damage, these kinds of things, as it's being transported back and forth between the nucleus and the cytoplasm. But the second thing is just DNA molecules are much, much larger than RNA molecules of a specific part of the DNA. That just means that the RNA copy, not only is it more expendable, and if it's damaged or lost, it's not a problem, but it's also smaller and easier to use. Remember, the protection of the DNA is exceedingly important. You have one copy of these DNA instructions. If something happens to them, the cell is at a loss for making any proteins that were carried in that instructions. So it is much better to make an RNA copy of the instructions, keep the DNA protected in the nucleus, and then the RNA can then make the protein, and then RNA is made to be a temporary molecule, then be destroyed until it may be needed again where it's made again. All right, so what is needed for transcription? This is an overview of just the things that are needed for transcription to occur. First of all, you're going to obviously have to have the DNA molecule that serves as the template to make the RNA strand. You also need factors that will initiate transcription, to start transcription. Those are called transcription factors in eukaryotes. It's a sigma factor in prokaryotes. Both of them will need the enzyme RNA polymerase in order to actually synthesize the RNA strand. You will also need RNA nucleotides to form the new strand. These are the units that are going to be used to build the new RNA strand. All right, for the stages of transcription, we have three main stages of transcription: initiation, elongation, and termination. We will now look at each of those stages and what occurs during those stages. We're going to start with initiation of transcription. Now we are going to focus right now on eukaryotes, but later we will discuss what is different with prokaryotes. All right, initiation of transcription. Mine's a little off the page here. So initiation of transcription. Remember, not all of the DNA is used to make the RNA strand, so only part of the DNA is going to be used. And in fact, not just a region, but only one strand of that region is going to be used. So only one strand out of the double helix will be used as a template. Not just one strand is always going to be the template. Different genes are found on different template strands. So some genes, if you are going to transcribe those, are going to be using one strand as the template. Another gene may use another strand as the template. Now, how are you going to determine what strand acts as the template then? There is a specific region known as the promoter region, which is on the DNA, and this is what determines which strand will be used as the template strand. The promoter region is where RNA polymerase will bind and initiate transcription. This figure just illustrates the previous concept that you can have different genes on different templates. Some genes are much shorter or much longer than others. So you can see on this particular figure we have quite a few genes here from gene A to gene G, located on different template strands and are of different sizes. Okay, now as we proceed, we're going to look at different terminology or use different terminology as we're going through this. Just like DNA, RNA strands have directional labels. They have a 5' and a 3' end. And just like replication that occurs with DNA, the RNA strand in this case with transcription is going to grow in the 5' to 3' direction, which means you synthesize a strand in the 5' to 3' direction. Just like replication as well, that means the template strand is read in the opposite direction from 3' to 5'. Additional terminology, upstream and downstream. This is related to the part where transcription is initiated. That's normally the reference point, although occasionally it's described or in reference with a different initial point, but normally if it's not stated, it's relative to the site where transcription has started. So any preceding DNA sequences before that transcription start site is referred to as upstream. Any DNA sequences that are following the transcription start site are now referred to as downstream. So you will have these two terms to kind of keep in mind, upstream and downstream. This figure just shows that in a representational form. If this is the transcription start site, preceding sequences are upstream, following sequences are downstream. Now what about the promoter region? The promoter regions are found upstream of the start site of the gene that you are going to be transcribing. The promoter is recognized by specific sequences that are contained within that promoter region. These promoters contain what are known as consensus sequences. Consensus sequences are sequences of nucleotides that are most commonly found at that specific location in the DNA or in the RNA as it applies. For most promoter regions in DNA, they have a consensus sequence that is very specific and it is conserved over periods of time. That's indicating that these are the promoter regions. So they are very consistently conserved over evolutionary time, and they don't really tend to have changes. One of those is known as a Tata box. Now the term is here, Tata box. Now it is named for the fact that it contains specific nucleotides, here again TAAA, and it is located roughly 25 base pairs upstream of the initiation site. Now on here, this particular figure shows the Tata box and the promoter. It also shows the gene that we are going to be transcribing. Now notice one thing about this figure. The promoter does not necessarily always have to occur completely upstream. Sometimes the promoter can also include part of the region where the gene is contained, as indicated by the dotted line there. The next thing is promoters, as well as the consensus sequences like a Tata box, are not always restricted to the same template strand as the gene is found on. So occasionally you will find parts of the promoter and the consensus sequence on the opposite strand of DNA as compared to the gene that you're going to be transcribing. Now this is because usually when you transcribe a gene, only a small region of the DNA is opened up to carry out transcription. Opening up here just means separation of the two strands of the double helix. What that means is the area of the promoter is probably often still very much associated with each other as far as a double helix. So they're very close to each other, and so both can serve as a promoter region. Now, as I explained before, the purpose of the promoter is to help bind the enzyme that's going to carry out transcription, which is the RNA polymerase. And one way it will do that is it will bind what are known as transcription factors. Remember, this is for the initiation of transcription. Transcription factors, that's their purpose, is to bind to the promoter region and then allow the RNA polymerase to bind. Now, why do these transcription factors have to bind to DNA during transcription? Why are they necessary? Why can't the RNA polymerase just bind directly? Again, mine's a little big here. All right, so the reason that you need a transcription factor is the RNA polymerase itself, which in eukaryotes is RNA polymerase II, cannot recognize the promoter region or initiate transcription on its own. It requires basically factors, proteins, to be able to point out where the promoter region is. Therefore, it requires these transcription factors to form or to start what's called a transcription apparatus. And that consists of the transcription factors, a mediator, and then the RNA polymerase, which will recognize those transcription factors. Okay, there are certain important transcription factors for you to know. These are transcription factors for the RNA polymerase II. So they are generally referred to as TF2, and on this nomenclature, that is the Roman numerals 2. Okay, so it's not 2 lowercase l's, it's transcription factor 2. And it's because it's associated with RNA polymerase II. Now, then they are subdivided by letters to indicate different types or different parts of the transcription factor apparatus. Part D, what its job is to bind to the DNA. And you can kind of think of it as part D, binds to the DNA. And its job is to recognize and be able to associate with the DNA. Now, as we're going to be able to see in a different lecture in the future, proteins associating with DNA is not as easy as it sounds. Remember, these transcription factors are proteins, and being able to bind to DNA, because they are not made out of nucleotides like DNA, is not as easy as it sounds. And proteins that bind to DNA need to have special characteristics in order to enable them to bind to DNA. All right, so the second transcription factor is called 2B. This assists in the selection of the start site. So its job is to point out, here's the start site for the gene. This is where transcription needs to start. And transcription factor 2H has helicase activity. Its job is to act as a helicase, is to separate the two strands of the double helix. Okay. Now here, it may be a little bit misleading, but just make sure you understand here, unwinding the DNA actually means separating the two strands from each other. Now, as I said, it is not always easy to have a protein associate with DNA. So here is a representation of a transcription factor with the Tata box ability to bind to the Tata box, so it can bind to the DNA itself. As we will see later, again, this is a specific kind of structure that it has. Notice that it has these kind of helixal areas, and we will look at those later because that's very important in binding to the DNA. All right, so once the transcription factors have bound, now on here, I've represented them as different transcription factors as the blue circles. Okay, so all of these. There's also the mediator, which we will explain the role of the mediator shortly. Once the different transcription factors have bound, the RNA polymerase II can now associate with the transcription factors and position itself at the promoter region. Now, other than the promoter, there are other regions of the DNA that can regulate the rate of transcription. Okay. Some of these are also known as a promoter, but with a different name. The promoter, where the Tata box is usually known as just simply a promoter or a core promoter. Promoters that have a different function that regulate the rate of transcription are usually known as regulatory promoters. Okay. What their job is to help initiate transcription, but also to regulate the rate at which transcription occurs. So we are going to talk about them now in this initiation part of transcription as well. There is another regulatory region which is known as an enhancer. All right, so regulatory promoters. Again, these are different than what is just generally known as a promoter or a core promoter. Their function of the regulatory promoter is just to make sure that transcription occurs at an appropriate rate, or the rate that transcription should occur based on the circumstances within the cell. Okay. In this particular case, proteins will bind to a regulatory promoter and their consensus sequences. So they will have a different set of consensus sequences, similar to a Tata box, but again, not a Tata box. And you will have proteins bind to the regulatory promoter at those consensus sequences, or they recognize those consensus sequences. Once those proteins bind, they will make contact directly or indirectly with that mediator, which is part of the core promoter or kind of regular promoter. And that interaction between the proteins at the regulatory promoter and the mediator at the core promoter, that will influence the rate of transcription. Now here, if activator proteins bind to the regulatory promoter, as the name suggests, activator, that will increase transcription rates. So again, increasing. If inhibitors bind, then that will decrease the rate of transcription. So again, if inhibitors bind at the regulatory promoter, that tells the mediator at the core promoter to decrease the rate of transcription. So here we have a representation of the regulatory promoter and the core promoter. Now, the core promoter would be in this area here. Okay. Notice that's where all the transcription factors are binding. Okay. And here is the mediator as part of the transcription factors at the core promoter. The regulatory promoter is almost always upstream from the core promoter. Here I'm showing activator proteins binding to the regulatory promoter. Those activator proteins are shown in green. They will make contact with the mediator. And basically that interaction is going to determine whether or not transcription occurs faster or slower. Now in this case, because it's an activator protein, it would occur faster or increase the rate of transcription. Okay, what about enhancers? That's the second type of regulatory region. And enhancers are sequences that occur in the DNA that will influence transcription, but they influence transcription at distant genes. So that is the key here. The difference between regulatory promoters and enhancers. Enhancers act at a distance. The gene that you are influencing is at a much greater distance. And in fact, enhancers can be thousands of nucleotides away from the gene that it's going to influence. Now you may be asking, well, how does that happen? But we'll go over that in a second. Okay. But the function is relatively similar. You will have proteins, for example, activator proteins will bind to the enhancer region. Then those activator proteins will associate with proteins at the promoter, and this means the core promoter, to increase transcription. Now this is a diagrammatic representation. Imagine if this is the promoter or the core promoter, and here's the gene that we are going to transcribe. The enhancer region may be located at a great distance from the promoter. Now in comparison, a regulatory promoter would be around here. Whoops, sorry, promoter. Trying to erase part of that. So the enhancer region could be at quite a distance compared to the gene and the core promoter. So how can that enhancer region influence the core promoter? Well, remember, DNA is flexible. So you can see here that the enhancer, by bending of the DNA in specific ways, can become physically very close to the core promoter and the gene that it's going to transcribe. Therefore, activator proteins at the enhancer, which is shown in green here, so the activator protein is binding to the enhancer, but now that is close in distance to the transcription factors at the regulatory promoter and can therefore influence gene transcription. Now, since this is called an enhancer, activator proteins bind to it, and this increases gene transcription. There is another regulatory region which is exactly similar in function to enhancers, except that they inhibit transcription instead of enhancing it. So those particular regulatory regions are referred to as silencers because they help to silence or inhibit transcription. Okay, so let's go through a summary of initiation, which we've been looking at. All right, for initiation, you will have transcription factors bind to the promoter. That will trigger the RNA polymerase to bind to the promoter region. Part of the transcription factors will allow a transcription bubble to now occur. Remember, the function of one of the transcription factors acts as a helicase. And then RNA polymerase can then start creating a new strand of RNA. That's its job, is to create the RNA strand. Okay, initiation is probably one of the harder parts of transcription, but now we're going to move on to elongation of the RNA strand. Now that we've initiated and started forming the RNA strand, how are we going to elongate it? All right, with elongation, the two strands of the DNA have been separated by the helicase activity of the transcription factors. So the two strands are separated in a relatively small region. Here it's only 10 to 20 bases on average are actually exposed. So there's not a large degree of separation between the two helical strands. Nucleotides are then added to the 3' end of the RNA strand. Again, you're building a strand from 5' to 3'. What will happen is, again, the energy is in the nucleotides that are being added. So as the nucleotide is added to the growing strand, 2 phosphates are cleaved from the nucleotide, and then that serves as the energy source for growing the RNA strand. And a little bit of trivia here, the rate of elongation in eukaryotes is roughly 60 nucleotides per second. Okay, this just shows a summary of elongation. Relatively straightforward. Here, as you're building a new strand, you just have to remember a couple things. First of all, you're building it in the 5' to 3' direction. So the new strand is always being created 5' to 3'. And the enzyme that carries that out is RNA polymerase II in eukaryotes. Okay, what about termination? Termination of transcription. Okay, in order to stop transcription, this is actually important, and it may be difficult to remember, but in processes like replication, the entire DNA is copied, so you don't have to worry about where to start and where to stop. Transcription, though, you have very specific start and ending points. So we went through this process about how you know where to start. Now we have to figure out how to know where to end. There is what's known as a terminator sequence in the DNA. And this, when it's transcribed into the RNA strand, becomes the terminator sequence AAUAAA. So there's quite a few adenines in there. Now, what will happen is, as transcription is occurring, the transcription will proceed beyond that terminator sequence. Even though it says it's a terminator sequence, think of it more like it's a signal indicating that the termination point is coming up at a certain distance ahead. So it's more like a warning that the termination part is coming up. Okay. Then what will happen is right behind the RNA polymerase will be a cleavage complex. It is made up of proteins, and what it is doing is it's following behind the RNA polymerase. Okay. The cleavage complex, once it gets kind of past the terminator sequence, it will find another consensus sequence. So, a different sequence beyond the terminator sequence. And that's just usually somewhere between 10 and 35 nucleotides beyond the terminator sequence. It finds that consensus sequence and it will cut it at that particular consensus sequence. That is why it's known as a cleavage complex. Now on here, this is also why this step above, transcription must proceed beyond the terminator sequence, is because the cleavage complex is following behind the RNA polymerase, and if the RNA polymerase stops at the terminator sequence, the cleavage complex behind won't be able to actually find the consensus sequence where it's supposed to cut. All right, so this shows a summary of termination. At the transcription stop site, you will have the removal of the RNA strand. The cleavage complex will cut it, and it will disassociate from the DNA. We've been previously talking about eukaryotic transcription. What about prokaryotic transcription? What are the differences between prokaryotic and eukaryotic transcription? All right, well, there's a lot of similarities, so thankfully there's not a lot of differences that we have to go through, but for initiation there are a few differences. First of all, prokaryotes are much simpler than eukaryotes. So prokaryotes do not use transcription factors. However, they use a protein called a sigma factor, which associates with the RNA polymerase and helps it to bind to the promoter region found in bacteria. So for initiation, sigma factor instead of transcription factors. For termination, this actually, I should indicate that really there's not really differences between prokaryotes and eukaryotes in elongation. Termination. Termination occurs by two different mechanisms. One is called row-dependent terminators, the other is called row-independent termination. So what is row-dependent termination? Well, in this particular case, there is a protein that's called Rho, and in the presence of Rho, which is the protein, the transcription will terminate because the Rho protein separates the RNA from the DNA and physically stops transcription. Once the RNA strand has been removed from the DNA template by the Rho protein, transcription has ended. What about row-independent terminators? Okay. In this particular case, there is a terminator sequence, and it is transcribed, and it generates a hairpin structure that stops transcription. Okay. Now, this hairpin structure occurs usually along with a string of uracil nucleotides, but this hairpin will stop transcription. All right, as previously mentioned, it is very important when you're dealing with transcription factors and other proteins that need to be able to associate with DNA, that these proteins be able to associate with the DNA in a stable manner, or at least stable for a period of time. But that interaction isn't as easy as it sounds. So what about the proteins and what makes them special in being able to bind to DNA? Proteins that are able to bind with DNA have structures on them which are called DNA binding motifs. That is the part of their molecule that allows them to associate with DNA. Okay, so the DNA binding motifs allow proteins to be able to associate with DNA. These regions on the protein are called the motif. And there are 4 different types as shown here. A homeodomain. This is a type of a helix turn helix. And so sometimes instead of seeing it as homeodomains, you can see them as helix turn helix. It's the same family or the same type of binding motif. Then there is a zinc finger, a leucine zipper, and a helix loop helix. Now please be careful. The last one here, the helix loop helix, even though it sounds like the helix turn helix, they are not the same. Okay? So just kind of keep that in mind. The name similarity does not mean they are structurally similar. All right, for a helix turn helix, on here, there's 2 different representations. On the right-hand side, which is B, is a kind of full scale model. On the left-hand side is more of a diagrammatic representation. The first thing you can see from B on the right-hand side is that these binding motifs allow the protein to be able to kind of closely associate with the DNA. If you look at A on the left-hand side, you can see the actual structure and why it's often referred to as a helix turn helix, which also includes the homeodomain. Okay. If you notice, the protein will have a helical region, and then it will have usually an area where there is a straighter region in between, and then another helix. Okay. And you can kind of see that on here. Here, a helix, a straight region, and another helix. Okay. So helix, turn helix, with the turn representing kind of the straighter region. Okay. Now, on this particular orientation, the helical part will insert itself into the major groove of the DNA. And that is represented with the asterisk. Okay, so that's the major groove of the DNA. As comparison, this would be the minor groove of the DNA. And this would be another major groove of the DNA. Major groove being a deeper area. Now you want your proteins to be able to associate with the major groove because it's going to form a more stable association because it's kind of deeper embedded there with the DNA. All right, this is the second type. This is known as a zinc finger. Now on here, if you look at figure C, it helps to show some of the characteristics. There are also some helical regions, which you can see. But you'll also see that there are green little flat arrows. Those represent what are known as a beta sheet. This is another kind of 2-dimensional or secondary structure of a protein. Now, also as the name suggests, zinc molecules, which here are represented as the black circles, are part of this motif. On here, it's the helical portions. If you now look at figure B, it's the helical portions that will now kind of associate with the major groove of the DNA, along with parts of the beta sheet and the zinc. What's interesting about the zinc finger is, unlike the helix turn helix, which was shown before, helix turn helix are individual. So essentially one helix turn helix can associate with one part or one major groove of the DNA. With a zinc finger, they can connect one zinc finger to another. As shown here, you can have finger 1, 2, 3, 4, and 5. So many zinc fingers can attach to each other. What that means is each finger can associate with a different major groove of the DNA, and then therefore it can allow the whole protein, if it has multiple fingers, to be able to associate with a larger region of the DNA. This can be much more beneficial because it makes a much stabler bond with the DNA. All right, now the last two, the leucine zipper and the helix loop helix, are shown together because they have very many structural similarities, a few differences, but they're very similar to each other. On the left-hand side is the leucine zipper. The leucine zipper is made up of two helical molecules, one in green and one in a yellowish color. And here they associate with each other at one point, but then they separate to straddle the DNA. And that's kind of the characteristic of a leucine zipper, and as we'll see shortly, the helix loop helix. By straddling the DNA, what it means is one part of the protein, in this case the yellow part, will situate itself on one side of the DNA molecule. The green part of the molecule will situate itself on the other part of the DNA molecule. So it straddles, or it sits on both sides of the DNA molecule. Kind of think of like a person's legs when they sit on the horse. One leg is on each side of the horse. That's kind of what happens with the leucine zipper around the DNA. Now a helix loop helix is very similar, with the exception of you have these little loop areas or straighter regions in between the helical part of the molecules. But it again straddles the DNA in much a similar way that the leucine zipper does. Okay, now we'll cover post-transcriptional modifications. Between making an RNA strand, which occurs during transcription, and the endpoint, which is a messenger RNA molecule that can be used for protein synthesis, there are many modifications that occur to allow it to become a messenger RNA. Now, first of all, a little bit of terminology. What comes out of transcription? So the RNA that leaves the DNA strand at the end of transcription is called a primary transcript or a primary RNA transcript. So some of the things that must happen is first, on the 5' end, a modified guanine is added to the 5' end to create what's known as a cap, or a 5' cap. Now here they show that modified guanine there. The second thing, on the 3' end, a bunch of adenines are added to the 3' end to create what's known as a poly A tail. The number of adenines is different, so that's why there's a small little n there, because it may vary. Sometimes there may only be 10, 12, sometimes there may be hundreds. So the number is going to vary. Now, why are they added? Well, first of all, a primary transcript that does not have these modifications at the 5' and 3' end are likely to be degraded. So these are an indication that the primary transcript is going to be used and needed to form proteins later on. So it's a signal, don't degrade me. Second thing, the 5' end is going to be needed to help position the ribosomes later during translation, and we'll see that. the poly A tail is needed to help remove it from the nucleus into the cytoplasm. Remember, this is a messenger RNA. It was created from DNA, which is in the nucleus. So transcription happens in the nucleus. The translation or creating of proteins is going to happen in the cytoplasm. You need to remove it from the nucleus to the cytoplasm, and the poly A tail plays a big role in doing that. Now the next component is you're going to have to remove what are known as introns from the primary transcript before it can become a messenger RNA. On this particular figure, the introns are represented in blue, and the key is over here. So the introns are represented in blue. They must be removed, and we'll go in a little more detail in a moment. So you must remove those introns. They leave, and you end up with only the parts of the transcript which are known as exons, here represented in red. Now at this particular point, when you have the cap at the 5' end, the poly A tail at the 3' end, and only the exons, you now have a messenger RNA. Now again, how are these introns removed? What is the process by which the introns are removed? Well, first of all, there is a structure known as the spliceosome. And I'll write that over here. This is a large complex whose job, along with some of these small nuclear ribonuclear proteins, will come in and form this lariat-like structure. It's just simply a loop. What it's doing is it's finding the parts, basically the ends of the introns. Therefore, once it kind of finds that part, it can remove that, and you are left with simply the exons. Now on here, notice that it's not just the removal of introns that's important. The spliceosome must also join the two ends of the exon together so that you get one continuous messenger RNA strand. You don't want to have it in fragments. Now in this case, why are there introns? It seems sometimes silly if you first think about it to say, why would you stick all of this RNA into a strand and then remove a lot of it? What would be the point of it? Why not just create the RNA strand that you want? Well, there are benefits to having this extra intron region inside there. First, we can look at this particular case. This is an example of a fibronectin gene. Okay. Now on here, there are different regions represented in different colors. Okay. Now we're going to be looking at two regions in particular. Here in green, denoted as E3B and E3A. Okay. In this particular gene, when you are going to make this gene and transcribe it, and then use that messenger RNA to create a protein, if you notice the fibronectin messenger RNA that comes from one particular cell, which is known as a fibroblast, here you have both of these green regions included, E3B and E3A. You can actually see them right there and right there. However, in a different cell type, which is known as a hepatocyte or liver cell, both of those regions are not there. In other words, inside the fibroblast, they act as exons. They are not removed. Inside the hepatocyte, they act as introns. They are removed. This tells you a couple things. One, that there is no such thing as necessarily a part that's always an intron or always an exon. It can be relative. Okay. As is shown in this case, those regions are sometimes an intron and sometimes an exon. But the other thing is it shows that in different cell types, they may use the molecule for slightly different purposes, meaning that in some, those regions may be necessary or helpful for their purpose, but in some other cells, they may not be. So what I'm going to do is, at this point, I'm going to stop the presentation, and we are going to pick this up in a different presentation to break it up into smaller pieces.